Muscle relaxation all starts with nitric oxide. Will nitric oxide bind to a receptor inside or outside of the cell? Let's look at the difference in electronegativity between oxygen and nitrogen. The electronegativity difference between the two atoms is less than 0.5, making nitric oxide both small and nonpolar and allowing it to diffuse directly through the cell membrane. Once it's diffused through the phospholipid bilayer, nitric oxide binds with its intracellular receptor, guanylyl cyclase. That activates the guanylyl cyclase, which is an enzyme that converts GTP, guanosine triphosphate, into CGMP, cyclic guanosine monophosphate. CGMP is known as the second messenger because it will now amplify the original message that nitric oxide sent. Protein kinases are a class of enzymes that play a key role in amplification of signals as they activate proteins by adding a phosphate group to them. In this pathway, the key protein kinase is called PKG for short, or CGMP dependent protein kinase. Hence, in order to be activated, CGMP must bind with PKG. Because enzymes are reusable, guanylyl cyclase can continue to synthesize CGMP, which continues to amplify the message. And now we're ready for the response. When muscles are contracting, the myosin protein in the muscle fibers has phosphate groups attached to it. PKG activates an enzyme called phosphatase that breaks down that phosphate group and thus allows the muscle, the myosin, to relax. Just as muscle cells need to be regulated in when they are relaxed versus when they are contracting, cells need mechanisms to both make and break down their second messengers. PDE5 is an abbreviation for a phosphodiesterase, an enzyme that breaks down CGMP into an inactive form called 5'-GMP. In the broken dog case study, Dr. M suggests giving the dog a PDE5 inhibitor. So how might that work? Let's take a look at it from the top. If an inhibitor is blocking the active site of PDE5, the CGMPs cannot be broken down, and thus they remain active to bind with the protein kinases and still bring about the muscle relaxation response.